was. So we are now officially in week 9. We are over midterm, and we need to talk about the neuromuscular junction. All right. So this is one of those core course objectives that it should be taught in every single physiology class, and specifically in your muscle physiology part. All right. So skeletal muscle, long and skinny, known as a muscle fiber. It is wrapped in endomyosin. It is not touching any other cell. It is an arrangement of a sarcolemma, so a plasma membrane on the outside, so those phospholipids. Embedded in those phospholipids will be some different proteins, some different glycoproteins, some different things that can interact with the outside environment, and molecules coming through the capillaries embedded in that endomyosin that might be leaking out and influencing things. So think about insulin can come in and bind to its insulin receptor on the sarcolemma, and it can change the muscle fiber's ability to take glucose from the extracellular fluid, which is coming from the blood, and put more glucose inside the cell. All right. And usually that happens after we've had something to eat and our blood sugar is spiking and our insulin hormones are going to elevate to help our muscles take up some of that excess insulin. All right. If we have been starving, again, glucagon is going to be made, and it's going to tell the muscles to not take insulin, not take glucose in, so that way the glucose that we have in our bloodstream is left in the bloodstream as much as possible for the neurons of our brain to use. Okay. And again, insulin and glucagon are working with the muscle to be a major place where we keep our blood sugar levels from getting hyperglycemic, too high, or getting hypoglycemic, too low. All right, so the plasma membrane, the sarcolemma, plays a role in that, okay? And that's where the capillary embedded in the endomyosin and its leaking contents into that area, that space, helps, again, the muscle have a role in whole body physiology for glucose regulation. All right, but other things in that endomyosin are going to be axons from neurons that are coming all the way from the spinal cord. And then if you go up the spinal cord, they're coming from the frontal lobe. That this is where the originating neurons are going to be determining, do I move my finger or not? All right, do I move my hand in which direction? And then the planning part comes even into the upper regions of the frontal lobe, closer to the forehead. All right, when a skeletal muscle is told to contract, the neuron that controls it, all right, so a skeletal muscle is tied to a neuron, and the neuron is going to have, it's going to sit in your spinal cord, and it's going to have a long axon that goes through the spinal nerve, all right? And so in my arm, that might be, it's part of the radial nerve, the ulnar nerve, and it's going to go, and it's going to spread out its little tentacles, and its little tentacles are going to maybe be, if it has 5 little tentacles, tied to 5 muscle fibers. Okay, so one neuron, depending on the number of tentacles, could be tied to controlling 5 muscle fibers. And each of those little tentacles push into a little area of the endomyosin and get really, 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 really close to the sarcolemma of the muscle cell that it's going to contact or control. Okay, now they're not technically going to fully touch. So that's why this is there's a space known as the synaptic cleft. But that is going to be the main way that the skeletal muscle is going to get information that's going to tell it to contract. And if it's not getting information, it's going to be resting. Okay, so the way a skeletal muscle is told to contract is by the neuron that controls it, all right? And it's told to contract through release of a chemical messenger that can cross the space, the synaptic cleft that exists between where the neuron's plasma membrane is versus where the muscle plasma membrane is. And that area, that type of synapse, is known as a neuromuscular junction. And it is a synapse, it is a connection place where a neuron can communicate with a skeletal muscle fiber. Okay, now some things to think about. This is a one-way communication, meaning the neuron is going to, under the conditions of electrical activity stimulating it, it is going to release a chemical. That chemical we're going to call, because it comes from a neuron transmitter. So the neuron is going to release neurotransmitters. The neurotransmitters will cross by exiting out through exocytosis, cross the synaptic space, and interact with proteins receptors embedded in the plasma membrane of the skeletal muscle fiber. Now, we're going to release molecules, and again, we're releasing molecules where some are going to try to float and they're going to float away, and some are going to like turn around and be like, let me back in, and then some are going to cross the divide and miss intersecting with the proteins, with the receptors. So the neuron's not going to release like 5 molecules and expect all 5 to go directly to the muscle membrane and bind to the 5 proteins. It's going to release like 20. And it's going to hope that, again, some of the ones that are going to like go lollygagging off to the side, they compensate for that. Some of them that turn around and start banging on their membrane going, let us back in, they're compensating for that. And then the rest of them might be, you know, out of 20 molecules, let's say 12 go down, go cross, and start interacting with the muscle. And out of those 12, you hope that 5 out of the 12 actually hit the proteins and cause a protein neurotransmitter interaction. Again, from chemistry, 2 molecules intersect, touch, and cause conformational changes in the protein molecule now feature that's going to lead to the muscle protein opening a doorway that lets molecules in the synaptic cleft, which is sodium ions, come into the cell, and it's going to alter the number of positive molecules outside versus positive molecules inside. It's going to alter that ratio, letting more positive in and cause an electrical event on the membrane. Okay? So, if I go to my whiteboard, let me stop sharing for a second. Where's my whiteboard? Nope. Of course, share whiteboard. So again, if you're going to draw the muscular junction here, usually the neuron will be kind of like some little petal or pod, all right? And remember, we'll draw it in red, your skeletal muscle is this long and skinny cell, okay? And because of all the myofibrils and all the sarcomeres, it has this striped appearance, all right? Okay. This entire thing is known as a neuromuscular junction, all right? The membrane, remember, of a skeletal muscle is known as the sarcolemma. Now, because the neuron is going to push into a designated part of that sarcolemma, okay, we are going to give this area of the sarcolemma where the proteins that are receptors for the neurotransmitter are going to most likely be made into a lipid raft. And again, this is all cellular theory. And the lipid raft is going to be a part of the membrane where these proteins kind of congregate. And they congregate here because this is the most likely area if they're going to act with their neurotransmitter, their receptor ligand. This is where it's going to happen. All right, so this part of the sarcolemma where the receptors for the neurotransmitter are going to be located, that is going to be known as the motor, because it's a motor neuron, the motor end plate, okay, like a plate you eat on. All right, now, just some general neuron talking to each other. This is a type of synapse, a place where neurons communicate with other neurons, neurons communicate with other muscles, and it could be skeletal muscle, smooth muscle, and neurons also have synapses where they communicate with glands to tell glands to secrete or dry up. Okay, this is the, again, one way. So this is called the presynaptic membrane. All right, that's going to be where your neurotransmitter is released. This membrane, our motor end plate, is going to be our postsynaptic membrane. It's going to be receiving the neurotransmitter, right? And the neurotransmitter typically lives inside vesicles at the terminal end, and through an electrical signal coming down the axon, it is going to cause, again, from chemistry and molecular biology, it's going to cause a change that leads to these vesicles merging into the presynaptic membrane, merging into the membrane of the axon and exocytosing out their contents, which is the neurotransmitter. Okay, and the neurotransmitter for a motor neuron, all right, a motor neuron and a skeletal muscle fiber is acetylcholine, which we abbreviate ACH. Okay, so these protein receptors are going to be acetylcholine receptors, all right, and they are going to be proteins that only are going to interact and bind to acetylcholine. And when acetylcholine is released in this synaptic cleft, all right, the space where the membranes exist, but they're not fully touching, all right, that is going to be the space that some of the acetylcholine is going to go out. Some of the acetylcholine is going to be moving in circles and trying to get back inside. And hopefully some of the acetylcholine is going to come and bind to its receptor on the membrane, on the sarcolemma, on the motor end plate of my muscle fiber, and cause that protein, that receptor, all right, to open a doorway, okay? And so this protein, if I draw it here, it's a protein that has a little spot where acetylcholine can bind. And when acetylcholine binds, it's going to open a doorway, all right? It's going to have this little flappy arm, again, just making, generalizations. And that opening is going to let sodium ions that exist, all right, sodium ions and chloride ions and amino acids and glucose and molecules exist in this water of the synaptic cleft and the water outside the cells of the endomyelin, the connective tissue, that's areolar connective tissue, okay? So when the doorway opens, all right, there's a little bit of sodium inside the cell, but there is a lot of sodium outside the cell. So there is a lot of positive sodium ions outside the cell, outside the muscle cell, okay? And there are few sodium ions. There's more potassium ions, actually, but there are fewer sodium ions inside the cell, okay? And so what happens is when acetylcholine binds to the protein and opens this door, sodium, which is in high concentration outside the cell versus low concentration inside the cell, sodium ions are going to want to come in through these channels into the cell. And when a positive ion that's normally high outside the cell rushes into the cell, it is going to cause an electrical event on the sarcolemma that's going to be able to travel down the length, down the width, and then because of our T-tubules, into the center of the cell and cause an electrical event that's going to release calcium from the sarcoplasmic reticulum across all the myofibrils through the length, width, and center of the cell. Okay? Hold on. Okay. So again, this picture gets a little messy. So let's go back to the slide that I I had up from our slides. All right. Okay. So if this is again now we should know these key terms. So the motor end plate. The motor end plate, yes, okay, is going to be this part down here. All right. this part of the sarcolemma of the muscle fiber. Okay? All right. The sarcolemma is the entire membrane of the muscle cell. So the sarcolemma is the entire membrane. The motor end plate is the part of the sarcolemma where all of these receptors are located that are in this position because the neurotransmitter, the ligand that can bind and cause these receptors to undergo a conformational change, is most likely to hit it and happen at this area, which is the neuromuscular junction. Right? The synaptic cleft is going to be this, again, extracellular environment that exists between the two membranes. All right? And remember, this synaptic cleft fluid, it is going to be a fluid that has things like glucose, sodium, chloride. It can have some amino acids. It can have molecules that come from plasma of your blood that have leaked out, and they're sitting in this extracellular environment until they're taken in by the cells and used for whatever cellular processes are needed to happen. But for the most part, the extracellular environment, the synaptic cleft, is going to be a sodium ion rich environment, and the sodium ions are going to be in high concentration. All right, so out here in this, again, blue area, we are going to expect high concentrations of sodium ions. And if we go inside the cell, we are going to expect low sodium ion concentrations. So these acetylcholine receptors, when acetylcholine binds to them and causes their doorways to open, and sodium is the ion that can fit through this doorway, so it's known as a sodium channel. It is going to let sodium ions from the outside of the cell where there is high concentration. It's going to let those sodium ions start to move inside the cell where there's a low concentration. And by letting a positive ion from the outside in, we are going to change the positive electron, negative electron balance. And that is what, in very simple right now terms, creates an electrical event that can now spread down the sarcolemma, across or around the width of the sarcolemma, and then through my T-tubules to the center of the cell. So all of the myofibrils and all the sarcoplasmic reticulums are going to get the message of the neuron just told us to contract, and all the calcium is released basically instantaneously across, around, and in the middle of the cell. And so all of the regulations on actin and myosin interaction are going to be removed at the same time, so the entire cell contracts and gives 100% effort. Okay? Again, our presynaptic membrane is the membrane of the neuron, and it is the membrane where the exocytosis of vesicles that contain acetylcholine, so acetylcholine can be released, is going to occur. The postsynaptic membrane is the sarcolemma, and again, the motor end plate where the receivers, the receptors are located. All right. Our neurotransmitter is acetylcholine. All right. Now, there are lots of neurotransmitters. Some of you have probably heard of dopamine, serotonin, GABA, GABA butyrate, or acetylcholine, norepinephrine. All right, so this is a very popular, and we're going to learn about it for another part of the nervous system, the parasympathetic nervous system, later. But acetylcholine is a molecule. All right, and when we break it in half, usually we break it as the acetyl component and a choline component. Okay, but when it's together, it's known as acetylcholine. Okay, now acetylcholine is made inside the neuron, and so it can be made and stored in vesicles. And again, don't worry about the specifics of how the electrical event leads to exocytosis. That's a very complex process also unto itself. But know that when an electrical event, when an action potential is going down the neuron, that means The neuron was told by the previous neuron in the spinal cord to contract, and the neuron in the spinal cord was told to the brain to contract. So the brain is telling basically the skeletal muscle fiber to contract. Okay, and acetylcholine is what crosses the divide, crosses the space between the two cells to turn an electrical signal on the neuron into an electrical event on the muscle fiber that releases calcium and lets actin and myosin across the cell, entire cell, and at every possible juncture imaginable inside the cell, lets all those actin and myosin cross bridges get formed, and with ATP present, contraction occur. Okay, now let me look at your questions. Okay, can you talk more what about acetylcholine is? Hopefully I've answered that. It's a molecule. It's a molecule that is made and hoarded in vesicles inside the neurons, inside, in this case, motor neurons that are going to release this neurotransmitter to communicate to skeletal muscle cells that are under the neuron's control that you need to contract. All right, so if we go back to my example, I have one neuron and its axon, maybe it comes out in T1, so it's part of right here coming out in my thoracic region, and it's going to be eventually going in and control some of the muscles in my bicep. All right, that one neuron might again have 5 little, ending axonal projections that are going to push in, and when told, all little 5 pods, so there'll be 5 of these, all right, they are going to all release acetylcholine in the synaptic space on the 5 muscle fibers that they control. And when those 5 muscle fibers, all within hopefully the same amount of time, get their acetylcholine receptors to bind to acetylcholine, the doors open, sodium rushes in, and now an electrical event travels down all 5, around all 5 sarcolemmas into the center with the T-tubules. All 5 of those muscle fibers are going to see calcium released, are going to see actin and myosin cross bridge, and then all 5 of those muscle fibers are going to contract. And it might be enough that it causes a little movement. It might be enough that it causes just a little twitch. It might be enough that it's just part of muscle tone. It's just part of, again, a little bit of the muscle staying active and pushing on my veins and pushing on my lymphatic vessels so fluid doesn't pool in my arm and heads back to my cardiovascular system. Okay, so that is going to be, again, where acetylcholine comes from. Okay, 1 through 7. I think they're trying to tell you 1 through 7 are the events that are happening. So 1 is that an electrical signal is coming down the neuron. 2 is that, just for making it as simple as possible, a series of events happen that lead to the vesicles to dock, okay, on the membrane. 3, if I think this is 3 is that the vesicles undergo exocytosis. Again, another process that involves some protein-protein interaction, some enzymatic and calcium and molecules to be present, but we get vesicle membrane phospholipids to merge with phospholipids of the presynaptic membrane. They merge, and the center contents, which were sequestered in the vesicle, are pushed into the synaptic cleft. 4 is the synaptic cleft activity of acetylcholine crossing over. And again, we release enough acetylcholine to make, compensate that some of it's going to try to come back into the cell, some of it's going to just float away, some of it's going to go across and miss the proteins that are the receptors, right? I think 5 is the receptors. The acetylcholine receptors are going to have an interaction with their ligand, acetylcholine, and that's going to trigger them to open. I'm not sure if this is 6. 6 is going to be that sodium ions are coming into the cell, and the sodium ions coming into the cell, as I've explained, is going to change the dynamics of how many positive ions are outside the cell, how many positive ions are inside the cell, how many negative ions are outside the cell, how many negative items are inside the cell. We have a balance. Well, that balance gets disrupted because sodium outside comes in, and that changes across the membrane this balance of positives and negatives, and it causes an electrical event. And that electrical event is what they show with the blue arrows here, and I've tried to show is going to then be an electrical excitement activity that's going to travel down and across and around and through, and then it's going to lead to contraction. Now, the neuron released acetylcholine, the neuron asked the muscle cell to contract. We need the neuron and the muscle to stop communicating. That is going to be where acetylcholine esterase comes into play. Okay, so when you learn about proteins, typically proteins that end in ASE are enzymes. This enzyme is an enzyme that takes acetylcholine and cuts it. All right, and remember, it's going to cut it between acetyl and choline. So this protein shown here in the blue circles, all right, it lives in and around the motor end plate. It lives in and around the neuromuscular junction. Some of these blue could also be potentially in the synaptic cleft hanging out. These proteins are what take acetylcholine and cut it at the acetyl and the choline connection. So they basically make acetylcholine, acetyl, and choline. So they cut it into two pieces. When acetylcholine is cut into acetyl and choline, it's no longer able to bind to the receptor on the motor end plate. So in effect, it is blocking the ability of acetyl and choline to trigger the muscle. All right, this is one of the mechanisms, and it's the most important mechanism that is going to remove the acetylcholine from the synaptic cleft and from the synaptic space and remove acetylcholine from activating the muscle. Okay, and so it's another reason, because it's sitting in the synapse. When I'm releasing the acetylcholine, some of that acetylcholine is immediately broken into pieces and bits because it's going to immediately hit the acetylcholinesterase, and it's immediately broken up. And so it never has a chance to even try to bind to the receptor because the minute it's released, it's snipped. All right, so acetylcholinesterase is the main mechanism that I remove acetylcholine from being actively able to bind to the receptor. And I remove it because I break it into pieces, and the two pieces are no longer the molecule that's capable of binding to the protein receptor. Okay, and so I think that's what one of the numbers is also representing. All right, so this is a very, very, very important part. How does a muscle know to contract, a skeletal muscle? It knows to contract because the neuron tells it. How does the neuron tell it? The neuron tells it by changing the electrical information that comes and originates in the frontal lobe. It takes it eventually from cell, neuron, neuron, neuron. The motor neuron that is going to have its little axons pushing into this, again, motor end plate, neuromuscular junction. It's going to take that electrical signal and convert it to a chemical signal. The chemical that is released is acetylcholine. Acetylcholine is going to be released in high numbers, and the hope is that some of the acetylcholine is going to make it to the receptor, bind to the receptor, and trigger an electrical event on the receptor that's going to lead to actin and myosin interacting. Okay? I remove that signal by getting rid of the acetylcholine and by acetylcholinesterase breaking it apart into its molecules, acetyl and choline, that are not able to bind to the receptor. All right? Now, this is, if there's 3 critical phases to get a muscle to contract, this is the first one. The way I get a skeletal muscle to contract is I tell it to contract. That means I have acetylcholine available. So if I, for any reason, do not have acetylcholine available and I don't have acetylcholine released, I don't have acetylcholine in the body, I will not get a muscle to contract. So remember, for spinal cord injuries, there's paralysis from the spinal cord injury down because I've broken the connection to my brain where the electrical signals originate to tell those muscles to contract. And so if I have an injury, everything below that injury, those neurons are never then getting the electrical signal to release acetylcholine. Acetylcholine is never released, so what do the muscles never do? Contract. And if the muscles never contract, then I never have the movements they can create, so I don't have the ability to walk. I don't have the ability to use my fingers, okay, depending on where the injury site is. All right. Another way I can block acetylcholine, again, there's a lot of molecular concepts. If I block the ability of the vesicles to dock, if I block the ability of acetylcholine to exit the cell, I can block, and botulism does this. So if you look at pufferfish, botulism can cause paralysis. Part of the way they work is they block exocytosis of acetylcholine. It's a complex thing. Don't worry about exactly how, but know that botulism toxin prevents vessels from merging at the motor neuron and letting acetylcholine in synaptic space. So I can't move because my muscles are not getting acetylcholine. Okay? My brain's probably saying, move, 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 but the neurons are trying to let the acetylcholine dock and they're not able to. Okay? And if that happens to my diaphragm and my diaphragm doesn't contract, I stop breathing. So that's why botulism, that paralysis of the diaphragm, can lead to death because I could stop breathing, and it's because I can't get acetylcholine to release from the neurons of the phrenic nerve and tell the muscles of the diaphragm to contract. All right? Another thing that we talk about or can see happen is if for some reason I massively trigger a bunch of my muscle neurons to release all of their acetylcholine. And again, sometimes they'll keep some of the vesicles in reserve, or some of the vesicles might not make it all the way to the synapse membrane and might not completely release and exocytose in one contraction. But if I keep sending a signal and I send strong enough signals that I completely deplete all the acetylcholine from the neuron, I'll get a massive contraction and then I'll get nothing. And I can't get the muscle to contract again because I can't get the muscle to contract again until the neuron builds back up enough acetylcholine that it can then do a secondary release. So what we're going to learn as an organ is if I have 100 muscle cells, I'm not going to ask the same 5 muscle cells to contract over and over and over and over again. I'm going to ask, if there's 100, I'm going to make 20 groups of 5. And I'm going to say group 1, contract. Group 2, contract. Group 3, contract. Group 4, contract. Group 5, contract. Group 6, and so on. And so by the time I get back to group 1, they've been able to, again, reset, clean up their synaptic cleft, reestablish the synapse, reestablish the vesicles in the neuron, reestablish the calcium and some of the inside of the cells place, and they're ready to contract again, and everything's in place. Okay, and so that's kind of how, again, neuron muscles interact. That keeps, again, my muscle having the ability to contract over and over again, because technically, when I ask the muscle to contract, I'm probably asking different groupings, different cohorts. And eventually those cohorts are taking turns and allowing my muscle to continuously to hold tension, but doing so with different muscles taking turns so they can reset and get ready for the next time they're asked to contract. Okay, so I want to leave this right here, because it's a lot of information, and hopefully everybody feels a little bit better about understanding the key of what is happening at the neuromuscular junction, why it's happening, and how it's regulated. So the key regulator is the acetylcholinesterase. So if I was to give myself a drug that broke apart or destroyed or blocked acetylcholinesterase, I would expect my muscles to be more twitchy because more acetylcholine would stay in the synapse, causing the muscle to stay in a state of excitement longer. If I give myself something that activates acetylcholinesterase, elevates their activity, I would expect my muscles to be, again, contracting and then relaxing and having a harder time to potential, or they would fatigue or stop contracting faster. Okay, so getting acetylcholine released, regulating the amount of time and the amount of acetylcholine that's present in the synapse is a big part of getting a muscle to contract. This is, again, out of 3 phases of what are the 3 key elements to get a muscle to contract. This is the first one. I have to tell it to contract. I have to tell it with acetylcholine and a sufficient amount of acetylcholine to be able to get an electrical event to happen. Enough sodium comes in that enough of the positive negative balance is disrupted that the skeletal muscle has an electrical event that travels across, down, around, and releases calcium across all of its sarcoplasmic reticulums. All right, do I have any questions? Okay, I'm going to stop